The lesson of today will go more into detail about cellular transport. So to understand cellular transport, yesterday we have seen in much more detail the plasma membrane. We have seen that it's made out of phospholipids, and that those phospholipids, because of their chemical nature with their hydrophilic heads and their hydrophobic tails, they naturally form a bilayer, so that the phospholipids automatically go with the tails to each other because they repel water, and the heads, they point towards the watery extracellular environment and the intracellular watery environment. So that we learned that those phospholipid bilayers, that they automatically form because of the nature and the chemistry of those biomolecules. So every cell has those phospholipids. They are produced, every eukaryotic cell, they are produced in the smooth endoplasmatic reticulum, and they surround every cell, also prokaryotic cells, okay? But they have other ways of producing them. And in that phospholipid bilayer, we have a lot of proteins, okay? And we have also learned that those proteins, they can swim around, let's say, in that phospholipid bilayer. They move around like little ducks in a lake on the surface, you know? And those proteins in that plasma membrane, they have different kinds of functions. We have seen that they can be, they have 5 main functions. Remember, they can be enzymes, they can be recognition proteins, they can be receptor proteins, okay? They can be transport proteins. And the last one, what was the last one? Connection proteins, okay? So that's where the 5, or let me check quickly. The 5 proteins we have in the layers. It is the recognition one, it is the connection protein, the receptor protein, the transport protein, and the enzymes, okay? Remember that, the 5 main functions of the proteins in the plasma membrane, okay? Today we are going to have a look how cellular transport works, because this is, like we have seen, one of the main functions of the plasma membrane. And if we go quickly revision of what the functions are of the plasma membrane, who can help me? The first and main function of the plasma membrane, what was it?
It was to regulate what comes in and out, no?
Yeah, very good, Santiago. It regulates the movement of substances in and out the cell, for sure. What else is the plasma membrane doing?
It's conformed by the phospholipids.
Yes, very good. It makes, in fact, the barrier between the inside and the outside of the cell. What else the plasma membrane doing?
It transports the cell.
Yeah, very good. That's what Santiago says. It regulates the exchange, but it does other things, not the plasma membrane. But when we talk plasma membrane, we include the protein. So basically, they have more or less the same functions as the protein. You know, they go hand in hand with each other.
It can also interact with other cells, right?
Very good, Emilio. It does communication, it does attachments between different cells, and it regulates all biochemical reactions, okay? And then I'm gonna share my screen. Then we're gonna talk about cell transport, you know, because in fact, cell transport is, it's the main involvement of that plasma membrane. It's very, very important function of the plasma membrane. So let me share my screen with you guys quickly to see what is the content in Canvas. There you go. So this plasma membrane and its proteins, we call that the fluid mosaic model, okay? You see that here, the fluid mosaic model, and it looks like this, okay? That's what we have seen yesterday. In a moment, we're gonna see this video about the cell transport, okay? And what we're going to see today is cell transport itself. Like I said yesterday, we have 2 main types of transport, which is the passive transport and the active transport. The main difference between both of them is that Active transport needs energy and passive transport does not need energy. It's as simple as that, okay? And passive transport, because it doesn't need energy, it's all powered by concentration gradients, okay? Like we have seen yesterday with diffusion, I put the liquid, the colorant in the water, and the high concentration of the colorant goes to the lower concentrations in the water until you reach balance. Then movement stops. It keeps on moving, but everything is mixed. So the driving factor of passive transport is a concentration gradient. It's the movement from high to low concentration. In active transport, you can move from low concentration to high concentration because you need energy. But we go there in a moment, okay? So let's first have a look at this video about cell transport, and then we go over the different types of cell transports we have.
Have you ever wondered what it might be like to be inside a cell? Imagine the genetic material, the cytoplasm, the ribosomes. You'll find those in almost all cells, prokaryotes and eukaryotes. Eukaryote cells, in addition, have membrane-bound organelles. All of those structures and organelles have different functions, but cells are not isolated little worlds. They do have a lot going on inside them, but they also need to interact with their environment. It makes sense that to keep a stable environment inside themselves, otherwise known as keeping homeostasis, they must have some control on what goes in and what goes out of them. A very important structure for this that all cells contain is the cell membrane. By controlling what goes in and out, the cell membrane helps regulate homeostasis. It's the homeostasis king. Let's take a look at the cell membrane. You could have a whole course on the cell membrane itself. It has amazing structure. It has signaling abilities. But to stick to the very basics, it's made of a phospholipid bilayer. Bilayer means two layers, so you have these two layers of lipids. Now, these lipids, they're called phospholipids, well, they have a head that is polar, and they have a tail that is nonpolar, making them quite unique. Some molecules, they have no problem going through the cell membrane, and they directly go through the phospholipid bilayer. Very small, nonpolar molecules, they fit in this category. They're a great example. Like some gases, oxygen and carbon dioxide gas, those are great examples. This is known as simple diffusion. Also, it doesn't take any energy to force those molecules in or out, so it's known as passive transport. Simple diffusion moves with the flow, meaning it moves with the concentration gradient. Molecules move from high concentration to low concentration. So when you hear someone saying that something's going with the concentration gradient, that's what they mean. They mean it's going from a high concentration of molecules to a low concentration of molecules. Now, remember how we said the cell membrane is actually a pretty complex structure? Well, one thing we haven't mentioned yet are proteins in the membrane, and some of them are transport proteins. Some transport proteins act as channels. Some of these proteins actually change their shape to get things across. Some of them open and closed based on some kind of stimulus. All of these are good things because it's helping with molecules that may be too big to cross the membrane on their own, or molecules that are polar and therefore need the help of a transport protein. This is known as facilitated diffusion. It's still diffusion, and it still moves with the concentration gradient of high to low. It doesn't require energy, though, so it's also a type of passive transport. It's just the proteins are facilitating or helping things pass. Charged ions often require a protein channel in order to pass through. Glucose needs the help of a transport protein to pass through. In osmosis, for water to travel at a fast rate across the membrane, it passes through protein channels called aquaporins. So these are all examples of facilitated diffusion, which is a type of passive transport and moves with the concentration gradient of high to low concentration. Now all the transport we've mentioned has been passive in nature. That means it's going from high concentration to low concentration. But what if you want to go the other way? For example, the cells lining your gut, they need to take in glucose, but what if the concentration of glucose in the cell is higher than the amount of glucose concentration in the environment? We would still need to get the glucose in, so it's going to have to be forced against the regular gradient flow. Movement of molecules from low to high concentration will take energy because that's against the flow. Typically, it's going to require ATP energy. A reminder that ATP, adenosine triphosphate, it has 3 phosphates, and when the bond for the last phosphate is broken, it releases a great amount of energy. Yeah, so ATP is a pretty awesome little molecule. ATP can power active transport to force molecules to go against their concentration gradient. And one way it can do that is actually energizing the transport protein itself. One of our favorite examples of active transport is the sodium-potassium pump. So that's definitely something worth checking out. There's other times a cell needs to exert energy for transport. We're still in active transport right now, but let's say a cell needs a very large molecule. Let's say a big polysaccharide. If you check out our biomolecule video, that's a large carbohydrate. Well, you may need the cell to fuse with the molecules it's taking in order to bring it inside. And this is called endocytosis. Think endo for in. Often this fusing of substances within the cell membrane will form vesicles that can be taken inside the cell. Endocytosis is kind of a general term. There are different types of endocytosis depending on how that cell is bringing substances inside. Amoebas, for example, rely on a form of endocytosis. In this example, pseudopods stretch out around what they are going to engulf, and then it pulls it into a vacuole. And there are other forms too, such as the fancy receptor-mediated endocytosis. This is where cells can be very, very, very picky on what's coming in because the incoming substances actually have to bind to receptors to even get in. Or pinocytosis, this allows cells to take in fluids. So to the Google to find out more details of the different types of endocytosis. Exocytosis is the reverse direction of endocytosis. Think exo and exit. They sound very similar too. Exocytosis can be used to get rid of cell waste, but it's also really important for getting valuable materials out that the cell has made. Want a cool example? Well, think back to those polysaccharides. Did you know that large carbohydrates are also really important for making plant cell walls? Cell walls are different from cell membranes. I mean, all cells have membranes, but not all cells have a wall, but plant cells do. And if you're going to make a cell wall, you're going to need to get those carbohydrates that are produced in the plant cell out of the cell to make the wall. So there's a great example of when you'd need exocytosis right there. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so that's it. That's a nice explanation, quick explanation about how that transport is working. Anyway, we're gonna have a look into more detail what it's all about. So certain things have already been introduced. I have to. So let's go back to the canvas content. So we have passive transport. So in the video, they repeated simple diffusion. I'm going to explain with another thing to go over it again. Then they also explained this facilitated diffusion. And what we still have to see is osmosis, which is not really explained in the video, but we go there right now. And then active transport is more simple. We will spend a little bit less time on it, but let's first have a look. Here you see another representation of what diffusion looks like. Can you see that, guys? Because I just switched screen. Can you see this diffusion thing here? Yeah, right?
Yes, we can see.
Okay, perfect. So again, What we have seen yesterday as diffusion, I did just the colorant in the glass of water because diffusion is happening inside the cell, but in fact, diffusion also happens over the plasma membrane, okay? The plasma membrane, we say that it's permeable, which means that it has pores or it lets things pass. So it's not completely a layer that doesn't let anything pass. Things can cross over the cell membrane, and we call it selective permeable or semi-permeable when certain things pass and other ones not. What you see here is water, oxygen, carbonate, and amino acid. And these molecules, we know that certain of them, they can pass from outside the cell, like you can see here, to inside the cell, and they cross over the membrane. Okay, how does that happen? Well, it's from high concentration to low concentration. So where do we have high concentration of water on this image? Camila? Out or inside?
Outside.
Outside, okay. Oxygen, do we have high concentration inside or outside, Ali?
Outside.
Outside, okay. So you understand high concentration means basically when there is more of that molecule on a certain site. So if I quick click play, you can see what diffusion does, and it starts moving, okay? So the molecules are moving from high to low concentration. And when does it stop? When will movement stop?
When they are equal?
Exactly, when concentration is equal, okay? So that's what diffusion is. It's the movement of molecules, small molecules in general, from high concentration to low concentration until balance is reached, okay? It's that simple. Then the second way of diffusion that we have seen is facilitated diffusion. It's exactly the same, guys, exactly the same with one big difference. Here you see a plasma membrane and the molecules can cross over the plasma membrane, but not all molecules can cross the plasma membrane because certain molecules are charged and certain molecules have a bigger size. They cannot cross over the plasma membrane. That's simple. So basically what happens is that there is a protein here in the plasma membrane, and the molecules that cross, they travel through a protein instead of crossing the membrane, and that's facilitated diffusion. It's the same, only the way how they cross is different. Simple diffusion, they can cross over the plasma membrane, through the plasma membrane better, and facilitated diffusion, they need a protein to pass, okay? Then there is this third thing, osmosis. Students are sometimes confused with it, but it's really simple. What osmosis is actually is diffusion, but it's diffusion of water, okay? And I like to explain it in the following way. Who likes to go to the swimming pool?
Me.
Okay, Emilio, if you are in the swimming pool and you're playing swimming pool volleyball, or I don't know what you do in the swimming pool, but anyway, assume that you're a long time in the swimming pool. What happens to you, Emilio?
Like your hands become like old people hands.
Yeah, indeed, they get wrinkled.
Yeah.
You all know what I'm talking about, that if you're a long time in the swimming pool, if you're a really long time in the swimming pool, they get so wrinkled that sometimes it even hurts. Did it happen to you?
Yes.
Okay.
Yes.
Well, do you have any idea why you get wrinkled?
Because like maybe because your cells balance the water that is outside with the one in your body.
Very smart, Emilio. Exactly. That's what happens. In fact, what's happening, if you're a long time in water, osmosis is happening, okay? And osmosis is nothing else than the diffusion of water. And what happens if you're swimming and laying in the swimming pool all day long is that your body starts to absorb water. Even if you're a long time in the swimming pool, you will also notice that you possibly have to go urinate more often. That's because your body is absorbing water through your skin. That's osmosis, okay? Osmosis is the diffusion of water. Okay, I make it a little bit more complicated right now. Emilio, do you like swimming in the sea?
Yes.
Yeah, you also like it. So you're in Cancun and you're in the hotel. And I also like swimming more in the sea because there are waves. I don't care, it's salty. But anyway, you have these hotels and then there is the swimming pool on the beach. And I always wonder, but why do the people swim in the swimming pool with the chlor and the chemicals when the sea is like just 10 meters further? Why do they do that? So imagine you're all in a hotel with the swimming pool and the sea, and you can choose to swim in the swimming pool and swimming in the sea. Now, my question for you is, where do your hands get more wrinkled, when you swim in the sea or you swim in the swimming pool? What do you think?
In the
I
swimming
don't know
pool.
because in both they have
No, in the
different
swimming pool.
chemicals.
Leonela is right. In the swimming pool, they wrinkle more.
Yes,
In the sea, they will also wrinkle, but not as fast as in the swimming pool. Do you know why that is, Leonela?
because the water from the ocean is like a bit more dense. I don't know how to say it, because it has salt and all of the ocean things.
Yeah, exactly. The ocean water is salty. So in fact, why you don't get wrinkled so quickly in ocean water is because that there is like a concentration of salt in the water. So there is still water being absorbed in your fingers, but not so fast. Okay, why not? Because there are salts, and the salts they in fact they prevent your skin from absorbing water. So to make a long story very simple, in fact, osmosis is the diffusion of water with a concentration gradient. So let me show you quickly. So in the sea, the water doesn't go so quickly through your skin because the salt is retaining the water on the outside, okay? But in the swimming pool, the concentration of salt in your blood and in your body is higher than outside. So the water will flow from with its concentration gradient. So it will be attracted to the high concentration of salt in your body, and that's why you get wrinkled. So if you look on my presentation here, that's basically what happens. You can see it here. Quickly, let me start sharing. So this is what osmosis is, okay? So let's take a certain cell, okay? In an isotonic solution, what you see here is a cell with 3 molecules of salt. Assume it's salt, the red things. And outside the concentration, there is 6, I know, but you have to see it in the volume. You know, it's the amount of molecules per volume. You know, so here it's 6, and on the inside it's 3. But basically, on the amount of volume, the concentration is the same. Okay, it's three molecules per milliliter, let's say, and here it's 6 molecules per 2 milliliters. Assume. So if the concentration inside and outside is the same, then there is no flux of water, no flow. So this is what we call isotonic solution, okay? The second case would be when the concentration outside is higher than inside. What happens is that water is extracted from the cell, okay? We call this a hypertonic solution because the concentration in the solution is higher than inside the cell, and water goes outside the cell with osmosis, okay? So osmosis moves in both directions. It just depends how the concentration gradient is, which concentration is higher. You can see that on my background as well. You have here on one side, you have water without sugar, and on the other side, you have water with sugar. And that the water with sugar will suck up the water, basically. And then here, the last situation, the solution has a lower concentration than the cell. So what happens is the cell will suck up the water, and you see the cell will become bigger, okay? Same things happen. We usually do an experiment in the classroom to understand osmosis. And what we do is, I'm going to explain you because we cannot do it right here, and we don't have the material because we need a digital balance. What we do to explain osmosis is very simple. I ask you to bring a potato, okay? And I ask you to bring what? Water, we get it from the tap. You have to bring a potato and salt. And what you do is you cut the potato in 2 pieces, and then you have to to weigh the potato, and each piece has to have the same weight, okay? So you take a piece of potato, and the potato is 50 grams, and you cut it into two, and then one half is 25 grams, and the other half is 25 grams. It has to be exactly the same. Then what we do is we put the potato in normal water from the tap, one piece, and the other piece, we put it in salty water. So we make seawater. We put 6 grams per 100 milliliters of salt, which is more or less the concentration of seawater. We put the other piece of potato in seawater. So my question for you now is, the potato in sweet water, Luis, what will happen? Will it still weigh 25 grams after half an hour or not? Will it weigh more than 25 grams or less than 25 grams?
More?
More. Yes, Leonela, it will weigh more. And why will it weigh more?
Because salt is heavy.
No, we're talking about the potato in sweet water. So the potato in...
Because
Sorry.
it absorb water.
Exactly, it's absorbed
Sugar.
water. Yeah, very good, Maria Fernanda. It absorbs water. You know that if you make caldito de pollo, the potato sucks up the water, okay? So the potato, after half an hour, that used to weigh 25 grams, it sucks up the water because of osmosis. It's osmosis. The water sucks up diffusion of water, sucks up the water, and it will weigh 30 grams, okay? Now, the potato in salty water, will it weigh 25 grams more or less than 25 grams? Who knows? Anyone?
I didn't hear, Mr. Can you repeat the question?
The potato that you put in salty water, and before you put it in, it's 25 grams, and then you wait half an hour. You can try this at home if you don't believe me. And then get it out after half an hour. Will the potato still weigh 25 grams? Will it weigh more than 25 grams or less than 25 grams?
The same?
More.
No,
A
not
little bit
more.
more.
Less.
I'm gonna say less
It's the same.
because
Less?
there's more salt in the potato, so
Yeah.
So the
exactly.
same.
No, Leonela, it weighs less. Because we make the
Less.
water very salty, and what happens is that the salty water causes the potato to dehydrate. The potato will weigh less than 25 grams because the water will move outside the potato to the salty environment. And that's what osmosis is. That's what you see on my slide right here. Okay?
So, Mr.
Try
Like,
it. If you don't believe it, try it at home. Yes?
it works like pressure, right?
It's not pressure, it's concentration gradient. The thing is that osmosis is the diffusion of water, and if there is on two sides of a certain membrane, in this case a potato, if the concentration of salts is different, it will push the flux of water, and that's what osmosis is. Okay, is that understood?
Yes, yes.
Think really the potato, the thing is that when we do it in the classroom with the potato, everybody is amazed because it does weigh less, guys. If you put the potato in very salty water, water is extracted from the potato, not absorbed. If you put it in sweet water, the potato will weigh less. So in the end, you have a potato that weighs 20 grams and 30 grams instead of 25, two pieces of 25 grams. We can, if we go back to campus, we can try if you want. Or try it at home. If you have a digital balance, you can try it and you will see there's always a difference. Okay, so simple diffusion, facilitated diffusion, osmosis. Let's now go to the active transport. Okay, active transport. So here, in fact, before I start with active transport, here, if you click read more, you have a very good explanation about simple diffusion, facilitated diffusion, what we have seen. And osmosis, you can see it in the presentation because there is not so much information about. Look, they only say water crosses by osmosis, always going where the salt is. This is exactly what I say, okay? And then, active transport. So active transport, we have, it's the way of transport, we need ATP. If you click on it, you can read about it. You have exocytosis and endocytosis, okay? And active transport is more simple, okay? I quickly want to show this with my presentation because it's a bit more clear than this. So this is the osmosis, active transport, we have it right here. Sorry, I have to look for it. Okay, here, can you see the presentation?
Yes.
Okay, so the first thing is, of course, active transport needs ATP, okay? And we have endocytosis and exocytosis. We have seen that already. Endocytosis is things going from outside to inside, okay? And exocytosis is the reverse, from inside to the outside. In endocytosis, we have three types. We have pinocytosis, which is drinking. We have the receptor-mediated endocytosis, which is basically that the molecule binds to the cell and then it's engulfed to be ingested. And phagocytosis is like cell eating, you know? It's the ingestion of liquids. You can see it here, okay? Pinocytosis, you can see it here, how the membrane is forming like a bulb. And then in second, it's gonna close the bulb. And 3, the bulb is inside. You see, you can see that here. 1, 2, 3. And that's what pinocytosis is, okay? This is an example of pinocytosis. It's taking liquid from the outside, from the interstitial fluid. 1, the membrane is closing, and 3, the liquid is inside the cell. So that's what drinking of the cell is. Here, number 2, we have an example of receptor-mediated endocytosis. So a molecule, a nutrient in this case, binds to a receptor. That's why we call it receptor-mediated endocytosis. It binds. Again, the membrane is bulbing, it's closing, and the third step, the molecules are inside the cell. And here you can see on the bottom, this is really how endocytosis works. In this case, receptor endocytosis, because this is an electron microscope example of how cells do this endocytosis, okay? It's very simple. You see that the membrane creates a coated pit. Again, it goes on the inside. The entrance is closing, and all of a sudden you have this vesicle on the inside. That's what endocytosis is. It's not for free. You need energy to do this, okay? And then here, the third thing is phagocytosis. So you see here, you see here a food particle. 1, same thing happens. It's closing, okay? 3, the food is inside. Endocytosis, very simple. Because you don't have to know more. You don't have to know how the energy mechanism is. It just is like taking things in the cell. What you do have to know, there's 3 different types of way of doing it. Phagocytosis, eating. Receptor-mediated endocytosis is when it binds to a receptor. And then pinocytosis is basically drinking. Okay, questions? Because that's really simple. No? And then here we have exocytosis. It's exactly the same thing, but the reverse. We don't differentiate in names, but here you see waste products. It goes in the second place, it goes to the membrane, and then the waste products are secreted. And here you can see that in an electron microscope, how that happens. So you have a vesicle and it pushes all the stuff out, okay? So that's it, guys. Really, it's that simple. This chapter, it's really not that difficult. But like I said, students sometimes, according my experience, they get a little bit confused with this osmosis, okay? I don't know why they don't get that. They sometimes, not all of them, of course, but some students, they get confused with endocytosis, with osmosis. And with osmosis, you just have to remember that this is the diffusion of water with a concentration gradient, okay? And a cell is doing cellular transport. It always needs to maintain homeostasis. Now we have some time left, and tomorrow you get a quiz about this. But before doing the quiz, you do this homework, please, and you can start doing that right now if you want. Then you don't have so much work. And what is about the homework is basically it's summarizing everything we have done today and yesterday, so you can do more tomorrow quickly a little quiz about it, okay? It applies the concept that you know. Here you see the plasma membrane. Okay, this is the plasma membrane. Here the proteins in the plasma membrane are represented pink, and you get some questions about it. What is homeostasis? Who can tell me?
The regulation of temperature.
Not temperature, Emilio. Homeostasis for a cell is to maintain a stable environment. So a cell, in order to function, needs nutrients. It needs like to have order and a certain concentration of all molecules. And homeostasis is this balance, is the correct balance of a cell in order to function, okay? It's in fact the balance between the inside and the outside of a cell that is a correct balance for the cell in order it to function correctly. But please look for a definition in Google and put the reference, but there you have it. What is selective permeability? Ivan, what is selective permeability? What is that?
I don't remember.
No? Selective means that it lets certain molecules in and others not. So the plasma membrane has selective permeability because depending on which cell you have, it will let water pass, it will let certain oxygen molecules pass, certain other molecules, but other bigger ones not. So selective permeability is like some of you can pass and others not. It really depends on the membrane, what passes and what not, okay? And the cell or the plasma membrane is basically this. We have seen that already. You have to give a definition that it's made out of phospholipids, that the tails are hydrophobic, the heads are hydrophilic. You have like proteins in it and mention the functions we have seen about the plasma membrane, okay? So here, complete the table, mark with an X or like a tachito in the correct column. So I can help you with this. I just want to help you guys because we have some time. Eh? So number 4, Ali, can you read this?
The process of maintaining dynamic balance or equilibrium within a cell.
What is that?
I think it is homeostasis.
Homeostasis, very good. Next one, Vanessa.
It provides the connection between a cell and its environment.
Okay, which one is that?
The cell plasma membrane.
Very good, it's the plasma membrane. Carlos, number 6. It's not there, Carlos. Andrea?
The characteristic of keeping certain substances outside the cell. I think it's selective permeability.
Yes, very good there. Selective permeability. Ana Sofia, number 7.
It separates prokaryotic and eukaryotic cells from the aqueous environment in which they exist. Plasma membrane.
Very good. You see, it's not so hard. Guillermo, number 9. No, 8, sorry.
The characteristics of allowing oxygen and glucose to enter. Homeostasis.
No, it's not homeostasis.
No. Cell plasma membrane.
Sorry?
Cell plasma membrane.
No, also not. You have one more option.
Oh. Yeah, selective permeability.
Yeah, it's selective permeability because cells, they have channels to let glucose and oxygen in. In fact, oxygen can travel through the phospholipids, and don't think they're just two random molecules. Cells, they are very, very dependent on both oxygen and glucose, because most cells nowadays are aerobic cells. You know, you have aerobic bacteria. Aerobic means that they need oxygen. So oxygen in many eukaryotic, prokaryotic cells, they can cross through the membrane freely, because otherwise the bacteria or the cell would die, because they need oxygen in order to function. Also glucose, because glucose is the molecule that gives the energy for the cell to operate. So the cell is not too picky about those two molecules to enter, because it needs it all the time. So it's selective, permeable for oxygen and glucose, because these are two very important molecules for any aerobic cell. Okay. Okay, so this is definitely selective permeability, because it's not going to question oxygen or glucose to enter it. It's say, look, please come along, because I need a lot of you guys. Next one, Emilio Marroquin, 9. Okay, that I see there.
Plasma.
Yeah, but how is your English? Because I see like a huge spelling error.
It's skipped.
No, you don't say
plasma membrane to the plasma membrane.
Okay, but what is the answer? But keep is wrong. You don't say keep.
Homeostasis. It's kept. Yeah, kept.
Yeah, they made a mistake. We made a mistake. It's kept. Yeah, it's kept thanks to the plasma membrane. It's homeostasis. Yeah, exactly. It's the homeostasis. It's separating. The homeostasis is basically keeping the internal environment different from the external environment is homeostasis. And then the last one who haven't, Andrea, please, I haven't heard you.
Which one?
Number 10.
Organelle present in all cells. Cell plasma membrane.
Yes, dear, very good. Okay, figure 2. So if you do this right away, you don't have the homework, guys. You just have to study this for the quiz tomorrow. So what do we have here? What is number 11? 11 is here. I don't know why they put L, but it should be 11. 11, okay, like this. So what is 11? What do you see? Sorry, what do you see here?
Is it a carbohydrate chain?
Yeah, very good. It's a carbohydrate chain. We have seen that especially carbohydrate chains, or we call them also glycol chains or glycoproteins, they often represent binding sites for certain molecules. So 11 is indeed the carbohydrate chain. Number 12, don't forget these carbohydrate chains are very often connected to a protein. It's not represented very clearly here, but usually they're connected to a protein. So 11 carbohydrate chain. 12, what is number 12?
Protein.
Yeah, it's a protein. And transport protein, to be more specific, you can recognize the transport proteins very easily because they go through the whole membrane very often. Okay, 13.
Hydrophilic.
Yeah, hydrophilic. The head of the phospholipid is hydrophilic because it's pointed towards the watery environment. Number 14, as a consequence, is what?
Transport protein.
The tail, no?
Yeah, it's the tail. No, 14 is the tail. It's pointing to this part here. It's the tail indeed of the phospholipid. Okay, very good. It's a hydrophobic tail. Or the non-polar tails. And then the last one, 15. What is 15? It's 15. It represents like the two phospholipids together. I mean, you don't see it that clearly.
So it's the double layer.
Yeah,
The B layer.
Yeah, the B layer. Okay, guys, don't forget it's a B layer. This is super important because this is like an amazing feature of the plasma membrane that it automatically forms these B layers. Do you know what else forms automatically B layers? You used to play with it all the time when you were a child.
Magnets?
No, not magnets. It breaks easily and you blow it.
A glow.
A bubble.
A balloon. A balloon. A balloon.
Who said bubbles? Soap bubbles, guys. The soap bubbles, soap molecules, they are not the same as phospholipids, but they behave similar. So if you blow a soap bubble, this is basically how a plasma membrane is working. A soap bubble is exactly the same. It's like it's a B layer of soap molecules automatically form. That's why bubbles are possible, okay? So if you think about the plasma membrane, always think about soap bubbles, because that's a very good analogy, you know? It's not the same, but it exists in a very similar way, you know? Normally we have in the same practicum of the potato, we play with soap bubbles, because if you take a soap bubble and you take like a droplet, like a dropper that makes drops, like a pincetta, you know, yeah, like a dropper, you can make a little water drop fall into a soap bubble without the bubble breaking. And the same way is how the plasma membrane works. So keep that in mind. Okay, so this is it for today, guys. I'm gonna leave this matching column A and B for you. Okay, it's not so hard. And then number E, it's also not so hard. Here, this is about table 3 is about osmosis. So these last 3 exercises, you can do it on your own because our class is finished. Do it, guys. Don't forget to do it before the quiz. Then I'm sure you're gonna do a fine quiz tomorrow. Okay.
When
So
is it for? Sorry.
Sorry.
When is it for?
Yeah, this is for tomorrow and the quiz is for tomorrow. So tomorrow we do a quiz about plasma membrane and cellular transport. So you have to know basically in the quiz that the plasma membrane is made of phospholipids. You need to know the function of the plasma membrane. You need to know the function of the proteins, and you have to have an idea of the 6 ways of transport, which is 3 ways of passive transport, which is diffusion, facilitated diffusion, and osmosis. And of active transport, we have like endocytosis and exocytosis. And then in the endocytosis, you have the pinocytosis, which is drinking, phagocytosis, which is eating, and the receptor-mediated endocytosis, which is molecule binding to a receptor. That's basically what the quiz will be about. And of course, you have to know what is a semi-permeable membrane. You have to know what is homeostasis and why a cell wants to have homeostasis. This is because otherwise it cannot function. It needs to keep this complex environmental, this complex internal environment in order to function. That's what life does. So that's basically what it's about. It's the quiz of tomorrow. I'm immediately going to share my presentation with you. If you want to have more details, you can find it in my presentation. You don't have to study every single detail of it. But it's a presentation that I usually have in my classes, and you can find all the explanation there as well. You can also use the content of Canvas to study for this. Okay, it's all in Canvas. You have the book. In the book you have available through Canvas. You can also find information from it. And like I said, guys, you always make your own notes for every quiz you do. Make your own little book with all the notes and summarize all the information there, that when you get to the exam, you don't have to go to all these different sources anymore. You just study your notes and you're good to go. You save a lot of time with it, and by making your own notes, you have, in fact, the most and the best information for yourself. So I said that many times already. I will keep on repeating myself because I really believe that's the way of studying, okay? So take care. If there's no more questions, we see each other tomorrow. Okay?
Thank you.
Have a good day, teacher.
Bye, sir. Thank you.
Thank you.
Bye.